This is a production of Cornell University.
So, today I'm going to talk about, like Joyce introduced, I've been actually looking back. I've worked on genome editing ever since 2009 as a postdoc, almost 10 years. I just cannot imagine, time goes so fast. So today I'm really going to share some results after, as PI, in the past few years, there's a lot of exciting, progress. I'll just share some results with you. So overall, my lab is really working on sort of genome editing, but we actually extend that more than genome editing. So we actually also engineer synthetic transgene activator repressors to turn on and turn off genes, which have their own advantage compared to existing technology. So, and we start a little bit, I don't want to talk anything about this yet, it's still very preliminary. We also want to do epigenome editing to change the epigenetic mark of histone modification, DNA methylation. But you have to keep in mind that in the end, even these epigenome editing, still you want to change gene expression. So rather, I think making synthetic transcription regulator is a more direct way. But again, giving today's topic and time, I actually going to limit my talk on genome editing. We can talk more if you're interested later. We can communicate on this topic. All right. So for genome editing, so to achieve that, it's rather easy to start because you have to start thinking about, can you engineer a very specific or tailor-made scissor? I call it magic molecular scissor to my daughter, for example, to explain that. Why it's magic? Because you can engineer that to recognize any DNA sequence as you wish in the genome, whether it's small genome in allogrossus or huge genome like in wheat. And so that has been a problem. We call sequence-specific nucleus because that involves a lot of protein engineering. How can you engineer? Because DNA sequence change letters, right? So that has been a big problem until lately when I start 10 years ago, or a little more than that, people have making some progress. But after you make double-strand break with whatever scissor you have, really that literally just introduced double-strand break. So DNA double-strand break is a very laser damage of DNA. So compared to other breaks, so it has to be repaired immediately before the cell cycle can be continuing, kind of replicating DNA can continue. So two pathways. One is NHEJ pathway called the non-homologous end-joining pathway, which literally just stitched together, eventually using ligase, like ligase 4 in plant, to ligase the broken DNA together. The pathway, in fact, I kind of argue is very precise. Okay, so you wouldn't make mutation, but if you make precise repair and it can be recognized by the same scissor again, it's going to cut again and making repair. Many rounds of this kind of thing go on, and eventually, and you will make error, make mutations. So that's how you introduce Indels, insertion small deletions. That's how you knock out gene. Many of you, I think, in the audience kind of use that technology to knock out gene nowadays, and for reverse genetics or other purpose. So, but another pathway, which kind of used minorly, in human cells as well as in higher plants, is called a homologous recombination or homology direct repair, HDR. This pathway typically is used in homologous chromosome or histochromatids if the cell is replicating, and to repair precisely the broken DNA. But we can take advantage of this repair pathway by providing a donor molecule which share homology on the left and the right. Then what's really in between the green stuff highlighted DNA sequence can be inserted, copied into the information, which really representing the precise genome editing. So when we talk about precise genome editing, we more or less referring to this kind of pathway. Unfortunately, this really pathway has been hard to achieve because it has been utilized minor in the cell. So when I first started, of course, this is a question we kind of want to try to overcome. And if you think about genetically, if you can block this pathway, NHEJ pathway, making some mutation of the gene in this repair pathway, probably going to shift the pathway toward this. So this is kind of one way when even as a postdoc, I did it. At the time, we are not really lucky as you guys have CRISPR. We only have zinc finger nucleus. So we engineer zinc finger nucleus to target gene in the background of Q70. Q70 and Q80 gene are just really the gene are involved in this NHEJ pathway. And now in the Q70 background, which means the NHEJ pathway is abolished. And we found actually, and we use the zinc finger to stimulate the repair of HDR. And indeed, we found the HDR frequency is jumped 10-fold higher. But even that is still single digit. And this is actually being done in protoplasts, which means somatic cell, not really germline. Whether we can achieve that, like a genome edited HDR plant is still open question. I will save that to the very late in this talk to mention this again. How can we achieve HDR plant, which are germline transmittable? All right, so start from zinc finger nucleus. And then later on, we have talent, which is again, the lab I worked with, Dan Volta's group, along with others, invented talent technology. But that really didn't stay very long, and later on being replaced by CRISPR-Cas9. And more recently, we have CRISPR-Cpf1. So for the sake of today's time, I'm really going to skip all my earlier work with zinc finger talent, rather give you some taste of CRISPR-Cas and CRISPR-Cpf1. So when I start my own group in North Carolina, ECU first, 2013, is my postdoc Levi joined the lab. So at the time, just happened to be CRISPR-Cas9 has already become a proven technology, even in plants, 2013. So but from there, we look at, as Levi, look at what can be done. We found a lot of things can be further improved. Researchers are still need a better tool. So that's kind of motivated us to do one topic, which is here. We developed a system called a PCR-free assembly of multiplex CRISPR-Cas system. The system rather simple. And basically what you end up having a sort of different step. You have a module of guide RNA, which you can basically start with a paired oligo, which you can order, and then you pair them, you insert that into a vector. Each vector is modular. We can, depending on how many sites you want to target. And if you have 3 sites, you have 3 guide RNA into different 3 modules, and using one single step of golden gate cloning, you put into together called a guide RNA expression vector. This guide RNA expression vector is also gateway compatible vector. So now we can utilize all the resources that plant research community have accumulated over the years of the gateway vector you all have in the lab. And then because gateway vector allow you to have different promoter, which is Pol-2 promoter, which can be inducible, also constitutive or tissue specific, right? So this promoter can now hook to drive different Cas9. For example, we have entry clone, which have all kind of Cas9, which are codon optimized for monocot, dicot, human, or anything, or Cas9 derivative for gene repression or activation. Now you can really have them all in this vector. Now, just 3-way gateway reaction, right? You can put them together. This is entry clone. These 2 guide RNA entry clone, Cas9 entry clone, the destination vector with different promoter. Now you're making single tDNA, deliver using typical agro transformation. This is how you work with this. And quickly, we have to demonstrate that when we publish papers, how it works. And we, for example, assemble 3 guide RNA and 2 targeting 1 gene to make a deletion, and 1 just making a single knockout in ROC5 gene. And in both cases, we can show T0 plant, you can have phenotype, which means biallelic knockout, very efficient. And then we also, this is my collaborator, close collaborator, Yong Zhang group in China. So we also want to, use that for other things. For example, what I showed you here is coding gene, protein coding gene. What about non-coding gene, like small RNA or microRNA gene, or long non-coding RNA gene, because that renewed recent interest in this kind of area, right? So we actually have evidence in this paper, if you are interested. So we show that even by introducing a small 1 base pair insertion deletion in microRNA gene, actually likely you're not going to abort the function. MicroRNA is still going to work. So the one safe way to actually knock out microRNA gene is actually conduct deletion by using double-strand break at this 5' and 3' end of this gene, and making a whole chunk of deletion. Now you can really make sure delete the microRNA gene. And we did that in mice. Is time up? Okay. All right. Okay. So good. We have 30 minutes. So all right. So deletion, now you have 60. You have 60%, right? Because 29 transgenic, you have 12 deletion happen, right? It's very efficient. And this one is 20%, but still it's very achievable, right? In rice, this is in rice. Making deletion, now you can knock out microRNA and to start its function. And then you ask me, what about other rices? People have been, kind of use that a lot. What about the plant I worked on? There's no even paper published about genome editing. How can I do that? Well, don't worry, because we have the tool, have a lot of Cas9. We have like 6, 7 Cas9, different coding optimized Cas9 you can test. And then, for example, this is a group, my collaborator in Poland did Rafa. So he used our system basically, had the visiting scientists come to my lab, did all the assembling and also other things learned, and we make all the vectors. And for example, here, just showing you 3, this is Arabidopsis codon optimized, Maize codon optimized, and plant codon optimized Cas9. And then you apply to carrots and knock out again the gene, F3H gene is involved in, this anthocyanin accumulation in carrot. So if you knock out, we start with a high accumulation line. If you knock the gene out, it becomes white, okay? So this quantitative looking at color cannot tell you how efficient different Cas9 you assembled, because literally you don't really add much work. Only on the last step of gateway cloning, you're just using different Cas9. It just doesn't really add much work at all. I mean, in the end, in the pipeline. But now you can test a lot of reagents. then he concluded this Cas9 optimized for allelopsis worked very well in carrots. So this is how you can do. And another example is collaborator Can1 group at Iowa State University. And so we, again, here just design two guide RNA to target 2 sites in the GL2 gene in Maize. And so she tests that 2 different constructs. And then we found that the efficiency is very high. In this guide RNA mediating 100% knockout. Every single plant is knockout. And many of them, if you look carefully, are biallelic knockout. Okay? And then another guide RNA is 90% knockout, also very high. So he basically, after she saw the result, she told me that, Yiping, we published a paper in Plant Biotech Journal last year reporting a Cas9 system for Maize researchers. But your system seems better. So she said, well, so that's actually good because the tool allow you to optimize the system. But we never really stopped, making better system. So when this Cas9 CRISPR-Cpf1 system has been reported in 2015, I become very interested for a lot of reasons. And just highlight a few of here. For example, the Cpf1 protein is unlike Cas9. Cas9 making blunt end cleavage 3 base pair away from the NHEJ PAM sequence. You all know that maybe. And then, but Cpf1 is different. It's just like zinc finger nucleus and talon that I have been working on. It's actually making a staggered end with a 5' end overhand. If it's making 3' overhand, which is better for HDR pathway, but still, the stick end may be really useful. For example, maybe we can use NHEJ pathway to really sort of inserting a donor DNA sequence to making precise gene replacement, allele swap, things like that. And that has to be done, in class. Not many people yet published. But also Cpf1, unlike Cas9 system, it actually only utilizes the CRISPR RNA, which is like 43 nucleotide long. For CRISPR-Cas9, we typically using single guide RNA, which is fusion of CRISPR RNA and the tracer RNA together, right? So much longer, like 100 base pair, 100 nucleotides. This is much shorter. And also, Cpf1 recognizes the poly-T, which is TTT, PAM, and which is a nicely complement to the NHEJ PAM of Cas9. So Cpf1 basically can recognize a more AT-rich genome or AT-rich sites. And also, it cut away from the PAM, which means if small mutation induced, maybe the guide RNA can still recognize site and cut again, which can stimulate further, if we pursue HDR, maybe can stimulate many rounds of cutting. So this is really very unique. So I become very interested. So again, my postdoc, we dived into really developing Cpf1. The first thing we tested, we just apply the same thing, how we express CRISPR-Cas9, we did the same thing with Cpf1. And we, in particularly, we express guide RNA with U3, U6 promoter. That ended up not working well. We just don't know why. We spent time troubleshooting. And in the end, we compared many systems, and this system come out very nicely. So the system we eventually worked out very well is, I call that dual POR2 promoter system, because we are using the same promoter of POR2 promoter, ubiquitin promoter for Maize, and to drive the Cpf1 protein. And what we tested is AS and ALB, because these 2 proteins just happen to be the one show activity in human cells. We just follow those things. And then we also use the same promoter to drive the CRISPR RNA. But here's the problem, because for POR2 promoter, that's unlike POR3 promoter, like U3 start with A, U6 start with G. It's very hard to define the first nucleotide of transcription. And also, we already, based on our failure with Cpf1 earlier, we have kind of already, I think, that this CRISPR RNA has to be precisely processed in order to retain very high activity. To do that, we use 2 ribozyme, which is hammerhead ribozyme, HDV ribozyme. For students here, ribozyme, is self-cleavage RNA, right? So just like enzyme. So now, with that, so you can imagine this transcript will be processed by self-cleavage after making RNA, and then this precisely processed at defined length of guide RNA. We're complex with, Cpf1 protein, and then act to recognize DNA genomically and making double-strand break. And indeed, this is case really working nicely. And here I just show you protoplasm data at 6 sites here, and where you can see ALB worked very nicely, but AS, half of them working, half not even working, so seem to be suboptimal. But ALB is robustly working. And then we look at what kind of deletion mutation make, and overall, both AS and ALB making over 95% are making deletions instead of insertions. Okay, they're mostly deletion. And in fact, deletion are actually sizes are larger. If you look at sizes, about on average is ranged from 6 to 13 base pair deletion, which is interesting because for Cas9, we typically in rice, we typically find like 1 to 3 base pair deletion. Okay, this deletion from CPF1 is much larger, and which can be explained by staggered end. That's more end processing after double strand break. So what's really exciting for us is really, we have to show whether we can make edited plants, right? So we're making agro-mediated transformation of making T0 rice plant. And excitingly, for the plant, we have surveyed and 100% knockout. Every single plant is knockout. And what's really astonishing is really that almost every single plant is biallelic knockout, which means already non-allele, okay, because both copy of chromosome, both gene has been knocked out because rice is deployed plant. So this is really exciting. So, and then, this is LB CPF1. And then, of course, other researchers, including Can1, are interested. So we collaborate. We show it's also working knockout gene in maize. So you can imagine you have now another tool, CRISPR-Cpf1 for genome editing. But the thing is, CPF1 not only to prefer AT-rich region, but the PAM, that the one working well, the AS and ALB nucleus in human cells are really preferring TTT PAM. If you search a genome or gene of interest, it's rather hard to find the triple T, right? So, it's really limiting you. And so then when I have a new postdoc join the lab in Shao, so she actually asked question, can we really making the CPF1 system more accessible, we can find more target sites in the genome, in the gene of interest, then it will become more useful, right? That's kind of obvious question. But then if you look at many of the, indeed, there's actually quite a few CPF1 protein sort of recognize only TTV PAM instead of TTTV. The V just stand, any nucleotide and not T, like ACG, okay, TTV. So, but the only FN CPF1 has been showing some activity in plants. And we look at literature, there's only one single site, which is actually edited by FN CPF1 is TTV. So all the site is actually TTTV. So although other researchers claim FN CPF1 recognize, TTV, there's no data backing that. So then we want to systematically look at this problem. So first we look at is, we just look at many sites, 10 sites here, which are TTV PAM. And then we design another different set of guide RNA, which are utilizing TTTV PAM. So for FN CPF1. Clearly, as you can see, the TTTV PAM, the sites are really show more activity, this is editing activity. So it's compared to TTV. But some TTV worked, some not really working, which kind of puzzling. But on the other hand, it's kind of suggesting that maybe hidden rule, which are dictating the activity of CPF1 recognizing TTV PAM. This is kind of make us to chase further how to make it robustly working at TTV site. But before that, we look at how CPF1 editing at the TTTV and TTV site compare. So this is the two TTV sites, and this is TTTV sites. Overall, they're making very similar profile. This is a deletion. This is kind of where the deletion occur, and this is deletion size. And if you look deeper, most of deletion actually are kind of repaired by microhomology of DNA. So then we also look at the specificity, because one big problem is it specific? Will there be any off-targeting problem? Any of us, involved using CRISPR-Cas will ask this question. So one thing one way to look at, instead of looking at homologous sequence in the genome, is just we can make mismatching guide RNA to target the same target site. And then we just making these mutating every two nucleotide, making mismatched guide RNA. As you can tell, most of the guide RNA abolish function completely, only at the very end, like the nucleotide 21, 22 nucleotide. And you actually see very similar activity, which kind of suggests, when we design CRISPR RNA for CPF1, especially for FN CPF1, maybe we don't have to design like 23 nucleotide alone. Okay, because 21 seems or 20 actually, maybe already working. But overall, I think it's still very specific because earlier mismatch or abolish function. This is at TTV site, this is at TTTV site, similar conclusion. So, but then we haven't really answered why FN CPF1 worked at some TTV site, some are not. So it's kind of still puzzling. So to do that, we look at, we basically using this approach. We search rice genome to find the exact same protospacer. Protospacer basically just target a sequence of 23 nucleotides. We just looking at rice genome to find the exact same site, okay, duplicated here and there, different chromosome, same sequence. And also both also have the CRISPR PAM, except one is VTTV. The reason is we are hypothesizing adjacent nucleotide may also play a role. impacting the function of CPF1. That's why we call it VTTV, because only difference is one is having extra T, one is lacking extra 1T. Otherwise, the whole sequence is same, but they are just located in different chromosomes, different sites in the genome. So then we let same guide RNA guided to recognize both sites, look at efficiency. So we look at that. I don't have to show you data because actually published just last month's paper. I just don't have time to go through. But overall, we found that, in fact, FN-CPF1 recognized most of VTTV, although a very different activity still, but they mostly fail to act on GTTA and CTTC sites. So if you want to design, if you want to target FN with, you try to avoid these two sites. That's kind of one conclusion. But how can you find another way to make sure you can target more sites in the genome besides just truncate the TTTV to TTV? So actually, Feng Zhang's group last year have shown they engineered some variant of AS-CPF1 as well as ARB-CPF1, and to recognize, for example, making the RR variants, just different amino acid change in the protein. Now the protein can recognize CCCC or TYCV PAM, just changed. So we kind of want to apply the same variant test in our system, whether it's going to work. And here's the result. And overall, yes, it's working. We actually also introduced the same mutation based on protein structure to FN, but we enjoyed less success. But overall, the red one showing is ALB, the R variant. Overall, it worked very well, recognizing the CCCC, 3 sites all working. But then many sites of TYCV, and then different variant, but all decent activity in protoplasms. But the FN variant are not really, that active. But nobody has ever published FN anyway. We figured maybe that's other rule, on that. And then in that study, they also showed the RVR variants, and then which recognize the TATV, and they claim also recognize TATC and TATG. We tested both, but we didn't really find it working in TATC. Rather, we found it showed 2 out of 3 sites, and both FN and ALB, this RVR variant can recognize TATG. So this is kind of the data from rice again. But you wonder, ask, what about can you make edits, stably transgenic plants, or even without transgene? We of course did it. Just this is summary, but I just don't have time to run through. But overall, I just want to give you the information. It's like whatever we see something working in the protoplast somatic cell, and then we can almost deduce whether it's going to work in stable transgenic plants or not. I even based our system is very reliable, very much correlated. For things not working, it's going to be 0%. But I just want to highlight here, for example, this ARB, the RR variant, recognize TTCC. We got almost nearly 100% knockout. It's very efficient if you're targeting those sites. But overall, after this work, and originally we start with the ARB and FN-CPF1, we know they can reliably recognize TTTV, which is like this many sites in the rice genome. And now with FN-CPF1, we defined which kind of VTTV PAM they can target. And now we quickly expanded to this many sites. And now we further engineered those ALB, the RR variant, you have this many of blue sites. And then you have the ALB RVR variant, recognize TATG. The overall, we kind of concluded with our new reagents, you quadrupled the sites you can target compared to originally you have. And we did a similar analysis in Arabidopsis, more or less the same. Now I want to shift gear to off-targeting because many of you are really interested in off-targeting, especially not only for this plant research, but also for biomedical research like human gene therapy, right, of application of CRISPR. So, but one thing I want to bring to you is here, for the same journal, Nature Plant, it actually published controversial papers. 2015, it published correspondence saying, it's very safe because, well, so not very safe, not much off-targeting found by whole genome sequencing of edited mouse. And then last year published another paper, like, okay, no, we found a lot of kind of collateral damage due to maybe Cas9 protein. Seems a lot of off-targeting, a lot of SNVs discovered in the genome of edited mouse. The beauty of both systems is they only sequenced one animal, which is being genome edited. I think the statistics, the sample size is too small if you all agree, right? So although they included a few parent, control, but even for this study, parent control the control animal become questionable. So that's why, if you all read the news last week, it retracted, which is no surprise, And Nature Medicine published editorial actually specifically addressing retraction of this paper. And they also published like 5 challenge paper to show why this paper has to be retracted, maybe something like that. The data analysis, how expression being designed, are not very good. So this paper is being retracted. And what's really interesting for me is when you read the editorial, the editor concluded, still to this day, there's still very little work apply whole genome sequencing to look at whatever animal organism has been genome modified, to look at whether Cas9 or Cpf1 have collateral damage in the genome. Not much work has been done. It's kind of like they conclude that. So, well, that's true or half true, but in fact, we actually, with collaborator, about 3 years ago, we already sort of thinking about this problem, actually, potentially, and about less than 3 years, but almost 3 years ago, we started thinking, how can we answer this question? Because this question has to be answered in a good way that then we can provide solid data to the research community or even to regulators like FDA or USDA agencies. So what we did is actually we designed a bunch of like 12 guide RNA for Cas9, and we collect them. We test those Cas9 protein for different activity in rice, ranking from very relatively low, like 25% of activity, based on how many T0 line you scored, how many are being edited. We score that efficiency. Okay, if you have 100 line, 25 being edited, that's 25%. But we also have Cas9 in some sites, 100%. Every single line is being modified, edited. And then we have Cpf1 protein, which is being modified at 100% because our system is very efficient. So, and then total we have 12 guide RNA for Cas9 and 3 guide RNA for Cpf1. So then what we did is we making a lot of transgenic modified plant. We T0 plant. And then we, based on the edits, we choose edited plant. We followed them to T0 because some of the edited plants still contain the transgene, still expressing the nucleus. Whether constitutively expressing nucleus will lead off targeting, that's another question. But for this experiment, the most important thing is not like getting these lines. It's really about getting the controls, right? This is the mouse study kind of lacked. So for us, we really include a lot of controls. For example, we know solely just having seed, from one generation to generation, by nature, you will have mutation. So that's why we actually prepare 3 generations of rice seeds from 2015 to 16 to 17, 3 generations of rice seeds from seed to seed. And we also look at tissue culture because the way we did genome editing is tissue culture is agrobacterial transformation in this case. And then we have plant which have agrobacteria infection, no agro. And we also have plant infected with agro contain Cas9 gene without the guide RNA. And also contain Cpf1 gene without guide RNA and Cas9 gene without guide RNA, all kind of control. And we sequence independent 2 lines each for these control. And these 4 lines each for these control, 12 lines here. So then many lines here. Total, we sequence about 70 lines total. And just to give you some idea, this is actually the analysis of control plants. You don't have to pay much attention. The point is, we try to use all 3 different software to look at to call out Indels, insertion deletion, as well as SNPs, and just to be sure. So this is just look at only use overlap for further analysis. And we did it for, this is just 12 control, but we did it for all the lines. And here's kind of some data. So the data, first of all, we are interested in whether we have mutation from seed to seed. Of course, if you collect seeds from seeds in rice, in our case, we found about 20 mutation, of Indel, SNV, SNPs, SNV, and also about 20 insertion deletion, half-half. Basically, on average, 40 mutations just from seed to seed, which is happen randomly. If you want to look at where they located, just randomly in the genome. And then we look at what the tissue culture introduced. Tissue culture, in fact, introduced about here is 120 SNVs, SNPs, and about here is like a little bit lower than 100, like Indels. It's varied because we have tissue culture only, or also agrobacterial infection only, and backbone only. And it seems agrobacteria resulting a little bit more in insertion deletions, but we don't want to claim that. We want to be careful because we still think our sample size may be still limited. But overall, you can tell, on average, it doesn't really seem to be, this on average, we show you a lot of mutation just in these control plants. You have to treat those mutation as background. In any plant being modified by genome, you're going to express this level of mutation, about 200 mutation in the plant you edited in rice, okay? And the question is, what about our really edited plant? So this is stacking the edited plant together. This red is just Cas9. The green is Cpf1. You compare to those backbone only. As you can see, on average here, so this is SNV, this is Indel. On average, they are more or less similar, okay? It's a similar level. And so if you look at whether you have 1 TD insertion, 2 or 3, because we happen to have some line even have 3 TD insertion, but I have to say overall 80% of transgenic plants only have 1 single TD insertion. So that's the beauty of agrobacterial transformation system. very limited number of copy number of tDNA. But nevertheless, we can find tDNA, not copy number doesn't really contribute in how many mutations we have. Okay, and then we happen to have a few constructs which simultaneously express more than one guide RNA. We duplex that to target 2 sites in the genome. And we also found that those even expressing duplicate guide RNA doesn't really seem to be having more mutation compared to single guide RNA plant. So really, there's no correlation. We did more correlation, but I want to skip those things. And but the conclusion is very clear. Then we look at now we are limiting to about 200 mutations every single plant we have, right? Now it's very easy. We almost know those mutations mostly due to tissue culture because we already found it in the control. But still, we can look at individually each mutation, whether really they are off-targeting due to Cas9 or Cpf1 or not. And I have to tell you, only one site from Cas9, we find evidence, we found a mutation indeed in those off-target sites. This is because we purposely designed one dirty guide RNA because we designed that way. And because we figured out if we don't find anything, it's rather disappointing, right? So we designed a guide RNA which has kind of off-targeting potential, like PCR primer, not really perfectly. There's a few other sites in the genome will be only 1 or 2 nucleotide mismatch. We know that. And indeed, those sites you find a mutation. Those sites can be luckily predicted by any computer software, right? You can predict that. But for you, it's good because now if you can avoid, the also conclusion we have is if you can make your guide RNA, your second closest guide RNA compared to your own target guide RNA, if the off-targeting guide RNA contain at least 2 mismatch compared to your guide RNA, you are very safe. It's very unlikely to be edited. This is based on our data, whole genome sequence data. And then we find another mutation, another line have mutation, but easily we figured out that's from parent line because there's no evidence of any similarity to the guide RNA. And all the mutation we discovered is like SNV rather than insertion deletion, because insertion deletion is the trademark of Cas9 or Cpf1 technology, mostly insertion deletion rather than SNVs. But these are mostly SNVs. We know it's most like a background mutation, and we just happen to be overlooked from parental line, even though we sequence so many parents, but still, that's variation among parents. You overlook some mutations. But this is only one site we overlooked. But overall, out of 15 guide RNAs, we only find 1 dirty guide RNA resulting off targeting. I think the conclusion is very clear. We think it's very safe. I don't want to say safe, but off targeting is very, negligible almost. So we then look at the T1 plants. We don't really find any. Just having Cas9 constitutively expressing in the plant. If the guide RNA is specific, we don't really have evidence of any accumulated mutation. So it's even safe to express them in the plant constitutively. So here now I've already covered a CRISPR-Cas9 system my lab developed and applied them in different plant species at high efficiency level. I also introduced you CRISPR-Cpf1, and more importantly, I shared the most recent data about off-targeting, and so far good. So it's very promising. It's really promising. I think that's really a promise in my title. Really, we can use them not only in rice, tomato, many other plants you can imagine. Luckily, recently the USD have regulation. They say we are not going to regulate these genome-edited plants like small insertion deletions, anything you can achieve by conventional breeding. So USD also makes that announcement recently. Our data really backing that kind of announcement. But I at the beginning I also introduced this how do you make this is another challenge, right? So how can you achieve HDR highly efficiently? And of course I continued the work. Many years ago I started because I want to achieve this high efficiency. But everybody in the audience may also agree Arabidopsis is really a great system, right, for study. But I have to conclude that it may not be a great system for genome editing. But we learned it in the hard way. And I'm going to share, I don't have time to share you data, but this is what we did is we really tried different reagents in Arabidopsis to really try to stimulating the genome editing in Arabidopsis. So this is work actually collaborated with my former mentor, Dan Voitas at Minnesota. So what we did basically, we tried a zinc finger nuclease, Talon CRISPR-Cas, and we expressed these reagents in inducible promoter and also constitutive promoter and even meiotic promoter. And we tried to knock in, because gene insertion by HR, we tried to knock in a gene with BAR, GUS, and GFP, different marker gene, different loci in the genome. And so what we really, and we even using viral replicants, replicate the donor DNA. And we tried many things, what we can. But in the end, what we found is we find many lines, some lines have very high evidence of HDR in somatic. So if you take harvest rosette leaf, if you evaluate gene editing by HDR, yes, it's present. Sometimes some line have 50% has been edited based on the viral replicant system. But then we harvest seeds from the plant to score them, whether they have event germinal transmitted. The answer is mostly no. So it's rather disappointing. We only have very little success. So what's really, so that's why we found a big disconnect between somatic and germline HDR events. It's disappointing. It's many years work. We felt, not only for the money we spend on that, for the people, for the work, but also for the community. A lot of people take things for granted. I think it's easy to do in alloptosis. We think we better report this, even a lot of negative data. So only way we figured maybe to report, get it published, maybe write a letter to editor, just because you have 23 supplement data are negative, maybe that's one way to go. But anyway, we write a letter. We think maybe that's the way, to report. I think I have some hypothesis, but I don't have time to discuss it. I think that's about DNA repair, about what kind of cell you're editing. But luckily, what we concluded here is maybe the better way right now is achieving genome editing. HDR is really to edit the plants like from somatic cell, like calli, and then regenerate the plants out of that, rather than harvest seeds from like alloptosis. Alloptosis is different because you use floral dip. So luckily, all the plants we are working on actually mostly go through tissue culture. I think that wouldn't facing this problem. That's kind of good side. But still, I believe all the plants, when we apply genome editing, more or less we have to overcome a little bit own problem, related to plant species, and which has to be worked out individually. And nevertheless, I hope you can convince that we are at the end of the beginning of genome editing, because we are at the end because we have, for most of you, you have enough tools to knock out the gene of interest if you only want to achieve knockout. But we are at the beginning of applying them to many plants, which are kind of exciting, I think. But another thing is about how can you make better product, right? So I think another limitation is can you get access to the elite cultivars, elite line like Monsanto or other agro big company they have, right? Then if you have that, maybe literally academic laboratory can do genome editing, making better product. People can grow them. Especially now it's not regulated. Maybe you can sell them in farmers market. Who knows? I mean, that's a lot of potential, right? That's why I think genome editing makes a lot of people very excited. But whether you can do that in germplasm, just quickly a snapshot to collaboration. So before I end, so one work I did with a collaborator, Yiping Qi's group. He has two labs. One is in China, one is in Australia. So with them, I'm being Zheng Yang in his lab. What we did is we knock out gene. He has been worked on many years, CSA gene. And this gene, if you knock out this gene, we use CRISPR-Cas9 in this case, and it actually, the trait is actually environment sensitive. So the mutant, for example, in this case, if you grow that in the short day, the pollen basically is infertile, cannot pollinate. But if you grow in a long day, and then it partly becomes fertile. Now you can basically switch the condition and making the pollen fertile. What's a big deal? But that's actually the technology behind a two-line hybrid rice system. Very nice, because hybrid rice conventionally rely on 3 lines. You have to have a maintainer line to give the one which are stellar, right? So that one can receiving other. But now we actually can making the same line, just switch condition, becomes stellar or instellar. So this is actually technology behind the 2-line. So this is one way we can apply this. The point I want to make here, we can directly making the edit in the elite line. They did it in 3 elite lines. Just show you one here. This is JY5B. This is the elite line farmer grow in the suburban area of Shanghai, for example. It's really the line, the germplasm people are using, right? So this is part of breeding. You can really apply CRISPR-Cas. And more recently, collaborator Yong Zhang we worked on is similarly, instead of one gene, we can target multiple QTR gene, right? Because seed trait, seed size, yield can be determined by many genes. But now we can multiple using all construct targeting many genes at once, and to look at not only target many genes, but also target those genes in elite lines. Okay, so here it's just like a 3, 2, 1 J, these 2 elite line anyway. So and then you can look at in this elite line, you can get single mutant, double mutant, and then triple mutant, and evaluate the gene knockout, because we work with alloptosis or the model plant takes things for granted, right? We think the gene of function can be translated easily to other crops. No, not the case. Even in the rice, even if you got gene in rice, in some other species, if you put in different elite line, maybe different. We have to evaluate that from breeding point of view. Indeed, we found variation for different trait. We evaluated it, even same gene. Okay, so this is again highlighted the importance not only work on EMT line, but you must almost have to test them in elite line to see whether really the performance of the trait you introduce really good or not. Okay, before I end, because like I said, I don't have time to do introduce any other tool, but we already published 3 papers about making activator repressor to regulate the genes. And this is 3 years ago. And last year, we published another paper on repressor based on Cpf1 strong repressor to repress gene of interest at transcript level. And a year later, earlier this year, we published another work. We repurposed activator, to make a stronger activator than the first paper to activate genes. We even repurposed the TAL system to making multiplexed gene activation. If you are interested, I encourage you to read this paper. I can make the resource available. In fact, all the plasmids are available at Epigenome. That's why I acknowledge that we try to make tools available to all the researchers even before publication. That's kind of what we do. And with that, I would really want to thank, first of all, startup from 2 universities and the company and the NC Biotechnology, Syngenta support my previous research. And I sometimes forget to mention all the collaborators in my slides. I go too fast, but I really want to acknowledge all of them. So every single, project I mentioned is through collaboration. Without them, not possible. And a lot of people in the lab did hard work. I will stop here. So take any question if you have. Thank you.
Hi. One of the questions in the field right now is what is sufficient genetic data to provide an answer for CRISPR-Cas9 or CRISPR-Cpf mutants. So when you think back about when people did EMS mutagenesis, it was generally felt that you saw a phenotype, you had to have at least 2 independent alleles. So the question now in the field is, is it enough to have 2 independently generated events from the same guide RNA, or is it?
If the mutations are different.
Or is it more appropriate to have, different guide RNAs producing these mutants? And with your sequencing data, you have a very nice way of addressing how we should proceed.
Yeah, I think to answer your question, I think first of all, either way will work. If you even design single guide RNA, it worked in your plant of gene of interest, making edits, but but of course it's gonna most likely introduce different Indel or whatever mutations, right? You have different events. If they all abort gene function based on what they got, and then they give you the same phenotype, I think that's fair to say it's strong enough compared to even the 2 independent knockout. Because if you knock out gene guide RNA, the end of the gene, maybe not fully abort function of the gene protein, right? So it really varied depending what kind of situation you're talking about.
The concern is really about off targets, though, and where you create the same off target mutation in multiple lines, and therefore, as a result, the phenotype could be.
Yeah. Along that line, it's really hard to address because the more is better in that case. More independent is better. And but if off targeting, is due to very much same homologous gene you are being edited because guide RNA is starting same sequence, maybe still you cannot really, Yeah, but overall, trying more is better. And I don't really think this is debating anything from conventional genetics, even EMS, honestly, because conventionally you have to do complementary. You have to make the wild type gene back to the mutant to show it rescue the phenotype. I think if you can show that, it will be even better, even with CRISPR-Cas. Yes.
Have you worked at all with any of the adenosine aminases, CRISPR base editors?
Yes, I do, but I don't have data here. So yeah, I work on that. Yes.
Do they work in plants?
Yeah, of course.
Okay.
they published. So yeah. So both C2T base editing and recently A2G base editing. Yeah, that reminds me. I have a couple master review on A2G. There's a lot of A2 people following A2G because David Liu published a few months ago about A2G base editing. But I have to say, overall, those base editing systems are lower efficiency compared to if you want to knock out gene, or compared to just using CRISPR-Cas wild type nucleus. Okay. Second thing is base editing seems to be promising things because it claims C2T or A2G change, but in the end, in that window of a few nucleotides, any C can be changed to T. Any A, you really are being limited. You have to screen a lot of plants, design guideline carefully. To me, it's not great yet. So that still have to, wait to work out window, narrow down, or broader window. That's different application. But my lab is actively looking at those areas. So.
But I think in quantitative genetics, we often want to just tweak things. We don't want to break the gene.
Yeah. I think base editing we have.
Treating
base editing will have a lot of application for promoter bashing for sure. There's a lot of other things. Or you want to screen a naturally evolving disease-resistant gene by stimulating changes, in across coding sequence of our gene. You can imagine, but to me, the system has to be made better. I'm working on that. So yeah, it's not that efficient yet. It's working. Yeah. Yes, Lee. Yeah.
Maybe I missed something here. For us to do transitional transformation, one to 20%. So what makes the transformation related, like you collaborate?
Mm-hmm. Oh.
Maybe the question.
The question, sorry, the question here is usually when you do transformation, you get like 10 to 20% transformation efficiency. How can you get even like 100% editing efficiency? Okay, here's a misconception. Basically, what we look at is we first look at the plants which are transgenic in this case, sort of select already, and among all the plants are edited, then we can say 100% edits. Yeah. If the one even don't carry transgene, we don't count because not transgene, not even have a reagent. So.
So
yes,
are there other Cas proteins on the horizon? Because you have Cas9, you have Cpf1 in your toolbox.
Yeah.
Flexibility. And I know it all starts out in other systems besides plants.
Yeah, that's okay. That's Cas13. Cas13 is RNA editing system. I have less interest in that because I have my own thinking, and because I have synthetic artificial transcription factors. I think those are really better to me, to use RNA cleaving, reagent. But then there's other company are developing. So I have heard about, but those are more or less like Cpf1. Yeah. Yes.
In one of your slides, you showed a picture of pear, which is a woody plant. So for many woody plants, woody perennials, they're highly heterozygous,
right,
and self-incompatible.
Right.
So can you think of a way to get around of this issue, like to get rid of the guide RNA at the end so plants are not transgenic?
Yes, there's a lot of ways. One way is you still deliver as DNA, and maybe you can find some edits, but just happen to be not stable. You can use you can edit without the tDNA integration, right? Deliver DNA, but the gene will be there expressing the reagent, but the DNA wouldn't be really inserting chromosomes and will be gone, after you're making a plant out. And then you can deliver with RNA, but RNA is unstable, right? That's why people are more using RNP, the ribonucleotide delivery. You purify Cas9 protein, and in vitro, just incubating test tube with guide RNA, and you bombard with gene gun or other way, or nanoparticle to achieve that. But to my understanding, those things are being worked out, but not efficient yet. Yeah.
We have time for one more question. Okay, if not, let's thank you.
Okay, thank you.
This has been a production of Cornell University, on the web at cornell.edu.